Amylase is an enzyme that catalyzes the hydrolysis of starch into sugars. Amylase is present in the saliva of humans and some other mammals, where it begins the chemical process of digestion. Foods that contain large amounts of starch but little sugar, such as rice and potatoes, may acquire a slightly sweet taste as they are chewed because amylase degrades some of their starch into sugar. The pancreas and salivary gland make amylase to hydrolyze dietary starch into disaccharides and trisaccharides, which are converted by other enzymes to glucose to supply the body with energy. Plants and some bacteria also produce amylase. As diastase, amylase was the first enzyme to be discovered and isolated. Specific amylase proteins are designated by different Greek letters. All amylases are glycoside hydrolases and act on I plus or minus -1, 4 glycosidic bonds. Classification: I plus or minus amylase. The I plus or minus amylases are calcium metalloenzymes, completely unable to function in the absence of calcium. By acting at random locations along the starch chain, I plus or minus amylase breaks down long-chain carbohydrates, ultimately yielding maltotriose and maltose from amylose, or maltose, glucose and limit-dextrin from amylopectin. Because it can act anywhere on the substrate, I plus or minus amylase tends to be faster acting than I squared amylase. In animals, it is a major digestive enzyme, and its optimum pH is 6.7 to 7.0. In human physiology, both the salivary and pancreatic amylases are I plus or minus amylases. The I plus or minus amylases form is also found in plants, fungi and bacteria. I squared amylase, another form of amylase, I squared amylase is also synthesized by bacteria, fungi, and plants. Working from the non-reducing end, I squared amylase catalyzes the hydrolysis of the second I plus or minus -1, 4 glycosidic bond, cleaving off 2 glucose units at a time. During the ripening of fruit, I squared amylase breaks starch into maltose, resulting in the sweet flavor of ripe fruit. Both I plus or minus amylase and I squared amylase are present in seeds. I squared amylase is present in an inactive form prior to germination, whereas I plus or minus amylase and proteases appear once germination has begun. Many microbes also produce amylase to degrade extracellular starches. Animal tissues do not contain I squared amylase, although it may be present in microorganisms contained within the digestive tract. The optimum pH for I squared amylase is 4.0 to 5.0. I cubed amylase. I cubed amylase will cleave I plus or minus 1 to 6 glycosidic linkages, as well as the last I plus or minus 1 to 4 glycosidic linkages at the non-reducing end of amylose and amylopectin, yielding glucose. The I-cubed amylase is most acidic optimum pH of all amylases because it is most active around penthouse 3. Uses: fermentation. Alpha and beta amylases are important in brewing beer and liquor made from sugars derived from starch. In fermentation, yeast ingest sugars and excrete alcohol. In beer and some liquors, the sugars present at the beginning of fermentation have been produced by mashing grains or other starch sources. In traditional beer brewing, malted barley is mixed with hot water to create a mash, which is held at a given temperature to allow the amylases in the malted grain to convert the barley's starch into sugars. Different temperatures optimize the activity of alpha or beta amylase, resulting in different mixtures of fermentable and unfermentable sugars. In selecting mash temperature and grain-to-water ratio, a brewer can change the alcohol content, mouthfeel, aroma, and flavor of the finished beer. In some historic methods of producing alcoholic beverages, the conversion of starch to sugar starts with the brewer chewing grain to mix it with saliva. This practice is no longer in general use. Flour additive: amylases are used in bread making and to break down complex sugars, such as starch, into simple sugars. Yeast then feeds on these simple sugars and converts it into the waste products of alcohol and CO2. This imparts flavor and causes the bread to rise. While amylases are found naturally in yeast cells, it takes time for the yeast to produce enough of these enzymes to break down significant quantities of starch in the bread. This is the reason for long fermented doughs such as sourdough. Modern bread-making techniques have included amylases into bread improver, thereby making the process faster and more practical for commercial use. Alpha-amylase is often listed as an ingredient on commercially packaged milled flour. Bakers with long exposure to amylase-enriched flour are at risk of developing dermatitis or asthma. Molecular biology. In molecular biology, the presence of amylase can serve as an additional method of selecting for successful integration of a reporter construct in addition to antibiotic resistance. As reporter genes are flanked by homologous regions of the structural gene for amylase, successful integration will disrupt the amylase gene and prevent starch degradation, which is easily detectable through iodine staining. Other uses. An inhibitor of alpha-amylase, called phaseolamin, has been tested as a potential diet aid. When used as a food additive, amylase is E number E1100, and may be derived from swine pancreas or mold mushroom. Bacillary amylase is also used in clothing and dishwasher detergents to dissolve starches from fabrics and dishes. Factory workers who work with amylase for any of the above uses are at increased risk of occupational asthma. 5 to 9% of bakers have a positive skin test, and a fourth to a third of bakers with breathing problems are hypersensitive to amylase. Hyperamylasemia, blood serum amylase may be measured for purposes of medical diagnosis. A higher than normal concentration may reflect one of several medical conditions, including acute inflammation of the pancreas, but also perforated peptic ulcer, torsion of an ovarian cyst, strangulation ileus, mesenteric ischemia, macromylasemia and mumps. Amylase may be measured in other body fluids, including urine and peritoneal fluid. A January 2007 study from Washington University in St. Louis suggests that saliva tests of the enzyme could be used to indicate sleep deficits, as the enzyme increases its activity in correlation with the length of time a subject has been deprived of sleep. History. In 1831, Erhard Friedrich Leuchs described the hydrolysis of starch by saliva, due to the presence of an enzyme in saliva, ptyalin, an amylase. The modern history of enzymes began in 1833, when French chemists Anselme Payen and Jean-François Section-Perses isolated an amylase complex from germinating barley and named it diastase. In 1862, Alexander Jakulowicz Danielewski separated pancreatic amylase from trypsin. Human evolution. Carbohydrates are a food source rich in energy. Amylase is thought to have played a key role in human evolution in allowing humans an alternative to fruit and protein. A duplication of the pancreatic amylase gene developed independently in humans and rodents, which could be suggestive of the importance of the gene. The salivary amylase levels found in the human lineage are 6 to 8 times higher in humans than in chimpanzees, which are mostly fruit eaters and ingest little starch relative to humans. References. External links. Molecule of the month February 2006 at the Protein Data Bank. Nutrition Sciences 101 at University of Arizona. Amylase at Lab Tests Online. Amylase, Analyte Monograph The Association for Clinical Biochemistry and Laboratory Medicine.